All right, this is the last of our lecture series on cell transport. It is entitled Active Transport. Here we see our objectives below. Please make sure you understand them by the end of the lecture. And as always, if you have any questions, you can ask your teacher in class or email them. So we're stepping into a whole new realm of cell transport, and the idea is that diffusion, osmosis, and facilitated diffusion are all types of passive transport, meaning they do not require energy. Why is that? Because they are always moving from a high concentration to a low concentration, and that's the natural flow of things, okay? Water will naturally move downhill, but think about it in the opposite direction. In order to get water to move uphill, can it happen? Yes, it can, but it's going to require a certain amount of energy in order to cause that movement, okay? So active transport is how we are going to move certain molecules that are going to go against their concentration gradient. And here's just a quick list of what we're going to talk about. Two different kinds of active transport that we focus on in ninth grade. We're going to talk about pumps and vesicles. There are two different types of vesicles there. Those are some vocab words that we're going to explore in this lecture. So first of all, pumps are going to require ATP, and the name makes you think of that. And what they're going to do is they're going to move molecules from a low concentration to a high concentration. So for example, sodium and potassium ions. These are very important in sending signals in your nerve cells, in your neurons essentially. And we're going to allow those ions to move through membrane proteins, but instead of just acting as a channel protein, we're going to have to pump these molecules across. We're purposely trying to push them in the wrong direction. So here's an example, okay? Here we're going to see, we're going to be pumping sodium ions. This is showing the result of what happens. There is originally a very high concentration of sodium outside of the cell, so we're going to pump sodium out of the cell, and we're going to take potassium into the cell. Why do we want to do this? Well, basically the explanation is see me in anatomy or AP biology class. But basically, in order to send an impulse, your cells need to maintain this difference in charge, okay? Because it's going to store energy, and so we need to keep a very, very high concentration of sodium outside of the cell and a very, very high concentration of potassium inside of the cell, and we block it then. So once we have that high concentration, we turn off all of the transport proteins. They cannot function. So it keeps those high concentrations where they are. But in order to get them high, we need to pump those ions. Another example of a pump would be hydrogen ions, okay? The lysosome is very important in the cell. We know that it breaks materials down, waste products, pathogens like bacteria, and old organelles. But in order to do that, it needs an acidic condition inside the lysosome at a constant rate. And so what's going to happen is we're going to need to constantly pump hydrogen ions into the lysosome in order to keep their concentration high. So again, we're moving from low to high concentration. It's going against the natural movement, so we need to put in some energy. Also, we're going to see the pumping of hydrogen ions play an important role in photosynthesis and in cell respiration, but there will be more to come on this in the next unit, so let's not get too far ahead. Sometimes active transport needs another type of assistance. So the pumps use transport proteins, but what happens if you have molecules that are even larger than transport proteins? What if, for example, you want to transport a protein itself? That's very, very big, and it's going to need assistance in moving. Energy is going to be involved in this, and what's going to happen is we're going to form vesicles, okay? So the vesicles that are going to form are these small sacs, and they're going to pinch off from the cell membrane or fusing with the cell membrane at the same time. So for example, if you are taking something into the cell, then it would be called endocytosis, and if you are making something leave the cell, then it is called exocytosis, okay? And so essentially, if something's coming into the cell, let me just draw a little diagram of what would be happening here. I have some animations posted online, so make sure you check those out, because sometimes in order to see a process, it helps to see it happening frame by frame all at the same time in one fluid motion. Here what I'm going to show you is, let's say there's a particle out here, a food particle maybe, or something that I want to take into the cell. What's going to happen is the plasma membrane is going to start bending in, okay, and it's going to dissolve this part here, and then it's going to allow, let's say this is my food particle, this little guy here. What's going to happen is it's going to bend in like that, and eventually the membrane is going to fold itself over until it seals off, and then this vesicle is free to move into the cell. Okay, so the cell membrane kind of folds in, pinches off, and is now inside the cell, where that material can go wherever it needs to go inside the cell. The opposite would be happening if we were trying to get something to leave. Okay, and so here what's going to happen is this vesicle, which is carrying, I don't know, let's say a waste product, okay, is going to fuse with the membrane. And when it fuses with the membrane over here, it's going to almost look the reverse of what it does in endocytosis. It's going to fuse with the membrane, and it's going to dissolve this little chunk of the membrane. So what can happen is that that particle can be expelled out. What have we not done? We have not let cytoplasm leak out. We have not lost any organelles or anything like that. We've done a step-by-step lengthening and then dissolving of parts of the cell membrane to flip the molecule from one side of the membrane to the other. So where do we see this happening? Okay, endocytosis, we're going to require energy. It's going to use vesicles, and we're moving from a low to a high concentration. This happens when we are taking in the bad guys. So your immune system is full of white blood cells, and their job is to eat foreign invaders like bacteria. So here is a really neat diagram. Okay, this guy here who looks like it has all these like fuzzy curly extensions coming off of it, this is a white blood cell. And what it is doing is engulfing these two bacterial cells. And you can see the vesicle wrapping itself around. Do you see how it almost looks like it's got like a little extra layer right here, and then this right here is also the same thing? That is the vesicle that is starting to form around the engulfing of that bacteria, right? These cells are called macrophages. And it literally means big eaters, okay? These are all coming from Latin roots here, so big eaters. So specifically, this type of endocytosis is called phagocytosis. The phage part of it here, meaning to eat. Cytosis is engulfing into a cell. So here we are engulfing whole bacterial cells. This is an important part of your immune system response. This right here, this is an amoeba. Okay, and we know amoeba have pseudopods, so they don't have any structure to them, but they bend their cell membrane. And what's going to happen is they're going to take this cell membrane and they're going to swoop it around, and they're going to trap their food, essentially, is what they're doing. Exocytosis is also active transport requiring energy, using vesicles, and is going to move from a low to a high concentration. An example of exocytosis we've already talked about in protein synthesis. So what's going to happen? Our protein is going to be made here, right? Let's say, and it's going to travel through the ER. And then how does it get from the ER to the Golgi? Right? Okay, it's going to move via a vesicle. And it's going to go into the Golgi through endocytosis. And then it's going to move through, and then it's going to be put into a secretory vesicle. And then that vesicle is going to fuse with the cell membrane, and then your protein product is going to be outside of the cell. Every time that you have a vesicle budding off of a cell surface, That is an example of endocytosis. It's how we can release proteins. And a very real-life example, okay, your pancreas is going to be responsible for making the hormone insulin, and insulin is required to help regulate your blood sugar. So when it produces insulin, this very huge, large protein, how does it get it out of the cells? It's going to need a vesicle. So here's actually a really cool example. You can see the vesicle is still formed here, okay? And it's fusing with this edge, which is the cell membrane, and then it dissolves that cell membrane, right? That used to be here, but now it's no longer here, and instead those insulin molecules are able to move out of the cell and into your bloodstream, where they're going to be able to do their proper signaling job. So insulin is an example of exocytosis. So at the end of all of these cell transport lectures, you should know diffusion, facilitated diffusion, osmosis, endocytosis and exocytosis, and different types of protein pumps. For each one of those examples, you need to be able to tell me, does it require energy? How does it move on the concentration gradient, meaning high to low, low to high? Does it require any assistance to move across the cell membrane or not? And also an example. We've given you lots of examples. So please make sure that you have an understanding of all of these cell transport mechanisms, and if you have any questions, let your teacher know.